Assalamu alaikum. This video will discuss documents 4 of chapter 9, sensory receptor and nerve impulse. Back in document 1, you learned about the resting membrane potential of a neuron, and in document 2, you learned how when this resting membrane potential is disrupted by an effective stimulation, an action potential is generated and propagates down the axon. Then in document 3, you learned about the characteristics of the action potential propagating in a nerve fiber. But in all three documents, the nerve fiber used is isolated from the animal, and the experiment is in vitro performed by electrically stimulating the fiber. Well, experimentally, the nerve fiber can be stimulated at any point. But in the human body, the genesis of an action potential occurs at specialized neuron structures. And thanks God, the stimulations that we receive are not electric. The human body receives many different stimulations from the surrounding environment. Those different stimulations are received by our 5 senses. Although our different sensory organs receive different stimuli, but they all have something in common. They all contain sensory receptors. Sensory receptors are like energy converters. They receive a stimulus in one form of energy and convert it into an electric one. And our body has all kinds of energy converters. These converters are the 5 senses: sight, smell, taste, hearing, and touch. All these senses have something in common. They all have sensory receptors, as we said. Sensory receptors detect changes in the environment. Sensory receptors are of different types, among which are mechanoreceptors, chemoreceptors, thermoreceptors, and photoreceptors. Mechanoreceptors respond to forces and movements. Chemoreceptors respond to chemical substances. Thermoreceptors respond to heat, and photoreceptors respond to light. Now let's see how these receptors work in our senses. For example, olfactory receptors are chemoreceptors found in our nasal cavity. These receptors are sensitive to chemicals that enter our nose. For example, if a vanilla molecule enters our nasal passage, it will bind to olfactory receptors and stimulate them to generate action potentials that will propagate to reach the brain where it's processed, and then you smell vanilla. You have 400 different olfactory receptors. That allows you to detect trillions of odors. Another example of chemoreceptors are gustatory receptors found within the taste buds of our tongue. Those receptors are stimulated by chemicals in food to generate action potentials sent to the brain to allow the perception of taste. On the other hand, our sense of sight uses photoreceptors that are sensitive to light. The different photoreceptors in the retina are stimulated by different light colors to generate action potentials that are sent to brain to allow the perception of images. Our sense of hearing, on the other hand, uses mechanoreceptors that are stimulated by the vibrations of the eardrum caused by sound waves. Within our skin, there are different types of sensory receptors as well. Some are mechanoreceptors sensitive to pressure and allows the perception of touch, while others are thermoreceptors that allow the perception of hot and cold. Don't worry, you won't have to study each of the previous examples, but you should know what are the different types of sensory receptors and to which stimuli do each type respond, as shown in document A of your books. So, whatever the type of sensory receptor is, you know now that it is a specialized neuron structure at which the genesis of action potentials occur. In the experiment of document B, a Pacini corpuscle is isolated along with the nerve fiber and placed in a physiological solution. A Pacini corpuscle is a mechanoreceptor found in skin. It's responsible for detecting touch, since it's sensitive to pressure. That is why the stimulus applied in this experiment is pressure. 3 recording electrodes are inserted at 3 different points of the fiber. R1 is inserted in the receptor itself. R2 is inserted just at the beginning of the fiber in a region that is referred to as the generation region. and R3 is inserted down the fiber. A pressure of 10 grams per centimeter square is exerted on the Pacini corpuscle. This pressure leads to a variation in the membrane potential of the receptor. This variation is called the receptor potential. And since the receptor potential exceeds the threshold of depolarization, the generation region records generating potential that in turn lead to the firing of action potentials that will propagate down the fiber. Now, if the pressure exerted on the Pacini corpuscle is increased as shown in document C, notice that the amplitude of the receptor potential increases as well, which leads to the generation of more action potentials. But keep in mind that the amplitude of all generated action potentials is constant and equal to 100 millivolts. Notice that no action potential is generated if the exerted pressure did not increase the receptor potential to reach the threshold of depolarization. This document reminds us that the nerve fiber obeys the law of none or all, and that in response to increasing intensities of stimulations, the nervous message within the nerve fiber is modulated by the frequency of the action potentials and not by their amplitude. Assalamu alaikum.